Thank you, David, for the really generous introduction. So I will introduce you on the basics of the CEST technique. Okay, Declaration of Financial Interest. Okay, so this slide summarizes what can be obtained currently with relaxation enhancers agent. Essentially gadolinium agent, but also iron oxide particles. So we can have beautiful perfusion studies, we can have beautiful angiographies, we can target high capacity, let's say, epitopes like fibrin. With some difficulty also one may eventually measure the pH. But we are far from what the biologists are used to do, because in the microscopic imaging they can recognize several epitopes in the same anatomical region. And so they work with this kind of multicolor image, reporting simultaneously on the presence of different epitopes. So we asked ourselves years ago if this can be done also by using MRI agent. Because gadolinium agent and any relaxation enhancers cannot do it because they act on the relaxation rate of water protons, and therefore the presence of a different agent in the same anatomical region will simply add one to another without having the possibility to distinguish the relative contribution. Whereas if one can make use of the fundamental properties of the NMR phenomenon, that is the chemical shift, through the generation of frequency encoding contrast agent, one can overcome such difficulty and can interrogate simultaneously the simultaneous presence of different agents in the same anatomical region. Of course, also the, let's say, image based on heteronuclei does a similar response. But the access to the large majority of scanners that operate on proton image, of course, make the CEST approach particularly valuable. So in short, what it is a CEST agent? A CEST agent is a chemical system that contain exchangeable protons and exchangeable protons or coordinated water molecule in the case of metal complexes that can exchange with the bulk water. Okay? So the exchange has to be in the slow intermediate regime, for which an high resolution spectra of such a system should result in distinct resonances for the exchangeable pool of proton and the bulk of water. Then the additional step is that one apply an RF field at the frequency of the exchangeable pool of proton in a way to saturate the proton that being transferred to the bulk water cause a decrease of the water signal and therefore a darkening of the compartment in which the contrast agent is contained. Now, one of the drawbacks is, of course, related that one can, when the exchangeable pool of proton is close to the water signal in the real cases, one can have a direct saturation of the water signal itself, and therefore it has been entered into the practice of the CEST experiment to, let's say, compare the transfer of saturated magnetization that occur when the RF field offset is set at the chemical shift of the exchangeable pool of proton with an analogous experiment in which the offset of the air field is set at the same frequency difference from the bulk water on the opposite side. So in this way, one can overcome the contribution coming from the direct saturation effect, as well as one can keep in account other contributions that are eventually present, such as those often related to the magnetization transfer. So this is the result, and so what an important parameter, of course, is that report about the efficient of a CEST agent is the saturation transfer percent. That is expressed by this simple relationship that relate the intensity of water upon the irradiation of the proper RF field in respect to the intensity of the water without irradiation. So this allows then to solve which are the basic issues that we say we cannot tackle with gadolinium or other relaxation agent. That it is to identify in the same anatomical region different agent, because now it's just important to shift the offset of the irradiation field in order to interrogate selectively the agent A or the agent B or the agent C and D, and therefore to represent in the image under different color the presence of the different agent. So this open a completely new route in MRI, in the contrast agent, because allow to approach the, let's say, the possibility to visualize different epitopes in the same anatomical region. So just to see which are the main determinant of the extent of the saturation transfer that one is going to measure. First of all, it is the exchange rate. I already specified that we have to stay in the slow intermediate regime because we have to avoid extensive broadening of the resonance that would require higher B1 values for the radiation field and then introduce SAR effect, as well that we have to avoid too fast exchange because they may lead to the coalescence of the exchanging protons signals. But the exchange rate, that is an important parameter that have to be properly modulated in the design of a new contrast agent. Another term is the relaxation rate of proton water itself, because it has to be as long as possible in terms of the T1, in terms of maintaining the saturation effect that has been introduced by the chemical exchange. Then, of course, the concentration of the system that generate the pool of protons that are responsible for the CEST effect, and the number of protons that are present, exchangeable protons that are present in the let's say, contrast agent. We will see that a lot of the development are related to optimize these parameters and in particular this N value. And of course, also as far as concern the instrumental parameters is the irradiation time, because we want to saturate as much as possible the pool of protons that have to be transferred. So this slide summarizes all the possible systems that are available for creating the CEST effect. So the exchange, the chemical exchange of a proton or of a water molecule, when we are dealing in particular with transition metal complexes or with other, let's say, supramolecular systems, are huge. And so one classification may be based, first of all, on diamagnetic agent and paramagnetic agent. Other kind of classification can be based on the endogenous molecule that generate a CEST effect in respect to extracellular exogenous contrast agents. So, and the number of molecules that can be exploited is really huge, And we still think that we are just on the top of the iceberg in terms of the application for the CEST agent, because still a lot has to be discovered. As far as concern, for instance, the endogenous CEST molecules, there is many group working in the field of amide proton transfer, because let's say this amide protons in protein presence inside the cells or in the extracellular matrix, endowed with a sufficient rotational mobility that are then kind of resonance that can be easily irradiated, allow, for instance, to characterize, let's say, the human brain tumors, that was shown in this seminal work from the group at Johns Hopkins University. Other small molecules, in this case, that can be delivered to the patient is represented by the simple glucose. Glucose contains several exchanging protons that can be properly irradiated, and therefore they transfer saturated magnetization on the bulk of water. This will be, I imagine, the topics that will be developed in the next presentation by Xavier Gollet, and so I just mention it here. Back to other kind of system, the paramagnetic agents are quite useful, and there is a lot of research going on, not yet, let's say, clinical trials about them. The advantage of paramagnetic system in respect to the magnetic agent is that the chemical shift of the exchangeable pool of proton can be very far away from the signal of water, thus allowing the exploitation of quite large exchange rate. And so we allow to transfer, let's say, significant amount of saturated proton to water. So, but if we look it as a whole, we can say that small-sized molecule, they contain a limited number of mobile proton, like sugar or amino acid. The sensitivity is an issue, because we have, let's say, we need a local concentration of mobile protons, which is in the range of tenths of millimolar. Whereas with the paramagnetic agent, we can go to lower values, you know. Eventually, if we exploit the exchange of coordinated water, that usually is quite a large chemical shift, we can go to in the order of hundreds of micromolars. Then increasing further the number of proton and going to macromolecular system, we can further improve the sensitivity for the magnetic agent in the order of the micromolar, and in the term of paramagnetic system in the presence of the magnetic molecule to form supramolecular system, just a few micromolar. But the system that in terms of sensibility, I would say, is the best, and it is becoming a reference in this field, is represented by the use of liposomes in terms of using the pool of exchangeable proton represented by the ensemble of water molecules that are present in the inner cavity of the liposomes. So now we can exploit a number of exchangeable protons that are of the order of millions or billions inside for each liposome. But one may wonder that the chemical shift of the water inside the liposome is the same or very similar to the chemical shift of the water in the extracellular compartment. And so in order to make to frequency encode the water molecule inside the liposome, it is necessary to introduce in the formulation a paramagnetic shift reagent in the inner cavity of the liposome, for which then the water molecule inside the liposome will have a chemical shift different from the bulk of water. And so we have the classical, typical spectrum in the high-resolution approach of a liposest, a CEST system. So now the sensitivity increases dramatically, and let's say for a simple spherical liposome, we can reach a sensitivity of the order of 800 picomolar in terms of the liposome concentration. And so when these are administered in vivo, one can differentiate a region in which they distribute, giving different color to the different kind of liposome. And this is the chemical shift of the water molecule inside the inner cavity of the liposome itself. Okay, so from liposome and further item of research that is currently under intense scrutiny is to say, well, if we exploit the water inside the liposome, why we could not exploit simply the water that is inside the cell? And in fact, this is possible if we internalize inside the cell paramagnetic shift reagent in an analogous way what we have done for the liposome. And so this can be easily done, for instance, for red blood cells, where simply an approach based on the change over the osmotic pressure. We can go from the isosmotic condition with the blood to an hyposmotic condition, the cell as well, and form a pore through which the agent may enter. And then we go back to the isosmolar condition. The red blood cell take again the usual discoidal shape, but now they have included agent inside. So according to the kind of agent we have internalized, we may have a different shift for the, let's say, exchangeable pool of proton, that when it is reported under what it is called the CEST spectrum or zeta spectrum, we look at the suppression of the water signal at the frequency of the water inside the cell. One can see that the effect is huge. In fact, according to the kind of magnetic moment, effective magnetic moment of the lanthanide ion, we may have a very high sensitivity, and just a small fraction of labeled cells can be detected in vivo, reporting, for instance, on the vascularity inside the tumor. This, of course, is not yet at clinical level, it's a result from research, but I decided to present it to show the huge potentiality that the system may have also in the field of cellular labeling. Finally, as I already mentioned at the starting of my presentation, one of the potential interest of a CEST agent is because they act as a responsive MRI agent. In a sense, they may report quantitatively on a parameter of interest without a prior knowledge of the concentration of the agent. It is important that inside the same molecule, or comparing molecules that have the same biodistribution, one may compare the extent of saturation transfer in respect to the parameter of interest. One of the parameters in which I was most interest is represented by pH. So it's nowadays possible to map pH. And in order to, let's say, have a system that could be immediately translatable to the clinical application, we thought of iopamidol, from whose structure it is possible to see that there are several kinds of protons that may be involved in exchange with water. We just limited ourselves to the amide protons, and inside the same molecule of iopamidol, there are two different amide protons, so with two different chemical shifts. And so the sensitivity of this that is usually used as a CT contrast agent may be applied in MRI, and in particular may be applied for mapping the pH. So if we look at the saturation transfer extent by irradiating one of the amide in respect to the other, the green and the red, we may notice that there is a different response of the saturation transfer level as a function of the pH of the two sites. Therefore, we may ratioing this values in respect to the applied magnetic field, and then have the possibility, when we measure experimentally the ratio in the saturation transfer from the two sites, to determine unambiguously the pH of the compartment. This has been done, for instance, to map pH change in the tumors region, or it has been applied for tumor, for instance, that was kidney, sorry, and this in tumor regions. And it shows, by the way, the high heterogeneity that one may have in different tumor in the same animal. And what it was interesting, for instance, in this work, it was to compare the FDG uptake by this tumor, by PET, with the change in pH that we measure, the average pH of the tumor. And it has been found that this inverse relationship. So this makes us rather confident that the mapping pH may be a very useful method to report about the glycolytic activity of cells, since protons have to be sent out in the extracellular environment where an agent like iopamidol can distribute in sufficient quantities. And this kind of studies actually has been brought already at the clinical level. This is a paper from the group of Marty Pagel in the United States, in which he investigated the tumor acidosis in the kind of breast tumor and mapping pH, and to see then to follow with, in particular, monitoring treatment and to see the pH. So this is my last slide, and I want to summarize the advantages that the CEST agent may have in respect to the classical agent. In particular, because they do not require the acquisition of a pre-contrast image. The pre-contrast image is simply represented by an image acquired when the offset frequencies is far away from the chemical shift of the exchangeable pool of proton. Then the very good, very important property is that more agent can be visualized in the same image, and finally that they may be used as a responsive agent, and I showed the example of pH, but of course other have been studied and reported. And I thank you for your attention.